Glycolysis is the first pathway in cellular respiration, which is an anaerobic process, meaning it occurs without the use of oxygen. High-energy electrons are also transferred to molecules of NAD+ to produce two molecules of NADH, another energy-carrying molecule. Taking place in the cytoplasm, a series of reactions occur that extract energy from glucose by splitting it into two 3-carbon molecules called pyruvates. In these reactions, two NADH molecules are made and two ATP molecules are made. The ATP are released into the cell for energy use, while the two NADH travel to the mitochondria, carrying high-energy electrons to the electron transport chain. The two pyruvate molecules created from the splitting of the glucose molecules go on to stage 2 of cellular respiration.
Before pyruvate can enter the next stage of cellular respiration, it needs to be modified. This period of modification is referred to as the transition reaction, a very short reaction that converts the two 3-carbon pyruvate molecules into two molecules of acetyl-CoA coenzyme A and carbon dioxide. The carbon dioxide is released and the acetyl-CoA moves to the mitochondria to enter stage 2 of cellular respiration, the Krebs cycle. NADH, formed by the reduction of NAD+, carries the high-energy electrons to the electron transport chain, stage 3.
Citric acid or Krebs cycle. Acetyl-CoA reacts with a 4-carbon molecule called oxaloacetate, forming citric acid, a 6-carbon molecule. Citric acid then gets oxidized to form NADH. This reaction causes the release of CO2, forming a 5-carbon molecule. This process happens again, releasing another molecule of CO2, forming a 4-carbon sugar. Then comes substrate-level phosphorylation. 4-carbon molecule undergoes another reaction, and its energy is used to form ATP from ADP and phosphate. The 4-carbon molecule gets oxidized 2 more times, forming FADH2 and NADH. This transforms the 4-carbon molecule back into oxaloacetate. The NADH and FADH2 produced in the citric acid or Krebs cycle are taken to the electron transport chain where oxidative phosphorylation occurs.
Now comes oxidative phosphorylation, which is an aerobic process, meaning it uses oxygen. NADH is oxidized into NAD+. It donates 2 electrons to the electron transport chain and H+ ions, which are pumped into the intermembrane space. FADH2 is oxidized into FAD. It donates electrons to the electron transport chain and H+ ions, which are pumped into the intermembrane space. As this continues to occur, a high concentration of protons is building up in the intermembrane space. At the end of the electron transport chain, the electrons are transported to the final acceptor, oxygen. Oxygen accepts the electrons and hydrogen ions from the intermembrane space and forms H2O, or water. Next, a protein called ATP synthase allows hydrogen ions to flow through it from a high concentration to a low concentration. H+ ions combine with ADP to form ATP. This process is called chemiosmosis.
Fermentation is another anaerobic pathway for breaking down glucose and allows glycolysis to proceed in the absence of oxygen by oxidizing NADH. In fermentation, the only energy extraction pathway is glycolysis, with 1 or 2 extra reactions at the end. In lactic acid fermentation, NADH transfers its electrons directly to pyruvate, generating lactate as a byproduct. Another familiar fermentation process is alcohol fermentation, in which pyruvate converts into acetaldehyde and CO2. Then, acetaldehyde is converted into ethanol, and NADH is oxidized to NAD+.
Antimycin A is an inhibitor of cellular respiration, specifically oxidative phosphorylation. Antimycin A binds to the QI site of cytochrome C reductase, inhibiting the reduction of ubiquinone to ubiquinol. in the QI site, thereby disrupting the Q cycle of enzyme turnover. It also will cause the disruption of the entire electron transport chain.